Welcome back to my channel Knowledge Chronicle.
Have you ever wondered how we create insulin in labs, grow pest-resistant crops, or even correct faulty genes in humans? Behind all these wonders is a fascinating field known as biotechnology, where science meets life at its most fundamental level, the DNA. Today, on Knowledge Chronicle, we'll dive deep into the principles of biotechnology, how scientists engineer life, clone genes, and produce life-saving products. Stick around, because what you're about to learn could blow your mind. Let's start simple. Biotechnology is the use of living organisms or their enzymes to make products that benefit humans. In ancient times, people made curd, bread, and wine using microbes. That's traditional biotechnology. But today, the word biotechnology has a more modern meaning. It refers to genetically modifying organisms to perform tasks on a larger and more precise scale. For instance, creating a test tube baby through in vitro fertilization? Yes, that's biotechnology. Synthesizing an artificial gene in a lab? Also biotechnology. Developing a DNA vaccine, like for COVID-19? Again, pure biotechnology. The European Federation of Biotechnology, or EFB, defines it as the integration of natural sciences and organisms, cells, parts thereof, and molecular analogues for products and services. That means using cells, genes, or their components, combined with science, to create solutions for health, agriculture, and industry. Modern biotechnology stands on two foundational principles. A. Genetic engineering. This is all about modifying DNA and RNA, the molecules that carry genetic information. The goal? To change how an organism behaves or what it produces. For example, inserting a gene that produces insulin into bacteria, so they start making insulin for diabetic patients. B. Bioprocess engineering. Once you have a genetically modified organism, or GMO, you need to grow it safely and in large numbers. This is done in a sterile environment, free from unwanted microbes, to ensure the desired cell or microbe thrives. Think of massive steel tanks called fermenters, where bacteria or yeast grow and produce antibiotics or enzymes. These two pillars, genetic engineering and bioprocess engineering, enable us to manipulate life in a controlled, beneficial way. You might ask, why go through all this trouble? Can't we just breed plants or animals the traditional way? Good question. Traditional hybridization mixes the genes of two organisms. While this can produce desirable traits, like a disease-resistant plant, it often brings along unwanted genes too. That's where genetic engineering shines. It allows scientists to isolate only the gene of interest, like disease resistance, and insert it without adding any junk genes. This gives precise control, like editing a sentence by changing just one word instead of rewriting the whole paragraph. So let's say we manage to insert a foreign DNA into a host cell. What happens next? Well, most of the time, the foreign DNA will just sit there, doing nothing, unless it becomes part of the host's chromosome. Why? Because only DNA with a specific sequence called origin of replication can replicate itself. So for a piece of alien DNA to multiply, it must be linked to a replicating DNA sequence and become part of the host's genetic system. This process is called cloning, making multiple identical copies of that DNA. Now let's talk about a breakthrough that changed biology forever. In 1972, scientists Stanley Cohen and Herbert Boyer created the first recombinant DNA molecule. They took a gene that made bacteria resistant to antibiotics, cut it out using special enzymes called restriction enzymes. These are nicknamed molecular scissors, and then pasted it into a plasmid, which is a small circular DNA found in bacteria. The glue for this job was an enzyme called DNA ligase, which joins the cut ends of DNA. This new DNA, a mix of original plasmid and foreign gene, is called recombinant DNA. When they inserted it into E. coli bacteria, the bacteria began making copies of the new DNA. This was the first successful gene cloning. A plasmid is a circular DNA molecule found outside the chromosome in bacteria. We use them as vectors, vehicles to carry foreign DNA into another organism. Think of them like, well, mosquitoes, which transfer malaria parasites into humans. Except here, the plasmid transfers a gene into a host cell. To summarize, the process of genetically modifying an organism has three basic steps. First, identification of DNA with desirable genes. For example, a gene for producing insulin or a gene that gives plants resistance to drought. Second, insertion of this DNA into the host organism. Using vectors like plasmids or even viruses, the DNA is inserted into bacteria, plant, or animal cells. Third, maintenance and transfer of the introduced DNA. Once inside, the DNA must replicate, stay active, and pass on to the next generation of cells. If all this is done right, we get a genetically modified organism that does what we want, like producing insulin, digesting oil spills, or growing faster crops. And that, my friends, is how biotechnology, using just a few molecules, is reshaping our world. From life-saving medicines, cleaner energy to better food, the principles of biotechnology help us design nature with precision and purpose. Did this video spark your curiosity? Then don't forget, your one like, share, and subscribe will help us create more valuable content. See you in the next episode on Knowledge Chronicle, where science meets simplicity.